So thank you very much. Unfortunately, I couldn't attend, but I'm happy to do this on Zoom. And I call this presentation a correlative budget approach because when we first started with our unit, we had very low budget and very simple instruments. And it was important for me to show that you can find some creative solutions. And even if you don't have this wonderful high tech that we all wish that we had, and some of us do, that you can do wonderful things with it. So I organized it with through organs, tissues, and cells. So let's go on. This is just a brief flash for the eyes to see what we have in our, currently in our unit as part of the Israeli node. So I was actually very happy that Sebastian mentioned the HRAM technique because it is something that I didn't know about. And also, this is something that we do not have in our facility. And that's my first tip, to find out what other techniques or microscopy methods people are using in your institute, because you might find some treasures that nobody knows about hidden in some lab that you can make use of. So this is such an instrument that I found. So this HRAM is a volume, a slice and view based method for small organs and whole embryos. And the samples are embedded in JB4 resin with additional eosine and acridine orange. If you can see here, I have a sample that looks very red. And this actually suppresses the fluorescence of the plastic, resulting in the in high resolution images of the block face. For this, yes. Okay, so the resin blocks are mounted on a motorized microtome, and the block holder comes to a rest at this photo position after each cut, and a digital camera, you see with the objectives, digital camera, automatically captures the images with 3 possible lasers or with bright field. So, okay, doesn't want to go. Back. Oh, computer is stuck. Trying to see what to do. Oh, computer is stuck. Oh, here. Okay, so here we can see the results from this HRM experiment. And we can see here a whole heart embryo, a whole heart of a mouse, an adult mouse. And we can see here wild type versus a deformed one, here from the outside and also from the internal part of the organ. You can see that even this bright field image is very, very good, and we can do some measurements inside. Here we can see a mouse embryo, a whole mouse embryo, and here it was correlated with the staining of the cardiomyocytes. So a very nice technique that I discovered we had. Now coming on to the tissues. Here we made use of the cryostat sections. Cryostat sections, old-fashioned, every university has one. Even in low-budget universities, you would have one of those. And this is one thing that you can really make use of it more than what you might currently do. So using correlating light microscopy and electron microscopy with using cryostat sections is a very good thing. It's also good for samples that are very fragile, and also like preclinical test before you go to more sophisticated methods. So here we used it in a study of susceptibility of lychee fruit to post-harvest water loss. To look at to measure water loss, normally they look at the outer layers of the cuticle and cell wall. So here we took a simple 20 micrometer section in the cryostat, and the only thing is that we collected them on cover slips and not on slides, on glass slides. And then it is dry. We took it immediately to the confocal. And here we made use of the natural autofluorescence of the tissues of the lychee fruit. And this is something I recommend with plants in general to see what kind of autofluorescence you are getting. So looking at the lignin in blue, cell wall in red, and cuticle in green, we were able to correlate in correlation with the SEM. It was they were able to measure whatever they needed there and correlated to the particular layer of interest inside of the fruit. Here again, still in tissues, and here we use a more sophisticated technique, multimodal correlative approach, volume approach, using confocal, micro-CT, and FIB. And here we use it to study the sperm storage organ morphology in Drosophila melanogaster female. This is an illustration, and we were looking at this sperm storage organ called seminal receptacle. And we use the combination of a confocal and micro-CT, first to localize where the sperm is, and then also to do the measurement, the right measurement, where and the location of where exactly we want to do the FIB milling. And we also use with the micro-CT itself a very a special way of sample preparation that allows us very high resolution. And we discovered inside the uterus of the female internal structural enfoldings that were not explored before, and we think that they are involved in the pathway of the sperm to the storage areas. Then with the FIB-SEM, of course, we did high resolution imaging and then segmentation. And out of this segmentation, we looked at an interesting fiber layer coating the tube, and also groups of vesicles that we then studied more in depth. And in this particular fiber layer, we went back to confocal, and here we used regular fluorescent staining of chitin, discovering that this is part of these fibers. If this is interesting for you, you want to know more, you can scan this QR. And lastly, I wanted to address the cells. So here we used a combination of confocal and field emission SEM for cytological slides. And here it was very interesting to study the chromosomal rearrangement and genome repatterning in Agilopus, type of wheat. We looked at clusters of tandem repeats that are here in red and in green, and the rest of the chromatin of these chromosomes are in blue. And what we did with it, we were able to do fish labeling that were fully mounted on a regular slide, but the slide was previously carbon coated to give it after better conductivity for the same. So normal fish labeling and a normal mounting, but the cover slip was sealed with removable rubber cement water-based glue. So after the confocal imaging, we were able to remove the cover slips under running water, dehydrate in ethanol series, and then air dry. Surprisingly, it worked really well. So after air drying, we can do another round of fish, or we coated the slides with iridium and went to the SEM. And in the SEM, they wanted to look at chromatin bridges between the chromosomes. And these undercondensed chromatin bridges, they could be thick double-stranded or single-stranded. And the thing is that the double-stranded, you could see quite well and okay in the light microscopy. But the single-stranded, more often than not, you couldn't see them. So the correlation with SEM was the only way that they were able to prove these single-strand bridges. And again, if you're interested in this project, you can look more into it there. And here, coming to a close. And here, basically, what I wanted to show today is that at any level of cellular organization, alongside the sophisticated imaging systems that we saw today and we're going to see today, that are expensive and may require some special expertise, there are some very good old-fashioned techniques like the cryostat sections that could be combined with a range of simple techniques present at any academic institute. And I would like to welcome you to consult with us if you want to find out how to make the most of your institute's treasures. Want to make contact? You can find us here. And thank you.